Dear students, I am Ramesh, chemistry faculty from Vidyamandir Metric Higher Secondary School, Uttangai. Let us discuss nucleic acids. And here we are taking the inherent characteristics of all species transmitted from one generation to next. So we are taking the inherent characteristics of all species are transmitted from one generation to next. One generation to next. And we are taking any characteristics is transmitted one generation to next. Then here we are taking that these are the responsible for that the chromosome present in a cell. So here the characteristics transmitted is responsible for chromosome present in a cell. Chromosome present in a cell. So nucleic acid. So the chromosome is made up of protein and that nucleic acid. Now we may discuss about nucleic acid, and the nucleic acids are two types. One is deoxyribonucleic acid. Deoxyribonucleic acid. Then it is known as DNA, deoxyribonucleic acid. Then another one, that is ribonucleic acid. Ribonucleic acid. So the ribonucleic acids are known as RNA. So the DNA and RNA present in the chromosome in a cell is responsible for the inherent characteristic transmitted from one generation to next. Now we are taking composition and structure of DNA and RNA. Now we are taking the composition and structure of DNA and RNA. So here, what are the composition of DNA and RNA? And here we are taking the nucleic acids are the biopolymers of nucleotide. So we are taking the nucleic acids. So nucleic acids are biopolymer of nucleotide. Now the nucleic acids are biopolymers of nucleotide, and the controlled hydrolysis, we are taking that the controlled hydrolysis of DNA and RNA, it will be produces one is nitrogenous base. We are taking nitrogenous or nitrogen bases. Nitrogenous base. Then the second one, it has the sugar component. So here it is a sugar. And the third one, it has phosphate chains. So we are taking that is phosphates. So here the controlled hydrolysis of DNA and RNA will produces these three components. One is nitrogenous base and sugar, that is a pentose sugar, and it has a phosphate. Now we may take one by one. The first one, it is nitrogen bases. Nitrogen base. Okay. And here the nitrogen bases, the nitrogen containing organic compound, it is mainly derived from purine and that pyrimidine. So these are nitrogen containing organic compound. Nitrogen containing organic compounds. And these are mainly derived from purine and that pyrimidine. So we are taking that it is purine bases, purine and also that pyrimidine bases. Pyrimidine bases. We are taking that the purine bases are, that is adenine and guanine. So we are taking that is purine bases. They are, one is adenine and the second one is guanine. So adenine and guanine are purine bases. Now we are taking the pyrimidine bases. Pyrimidine base. So pyrimidine base are, there are three types of pyrimidine bases. One is thymine and cytosine, uracil. These are the three bases. So we are taking, it is a cytosine, then it is thymine, then we are taking uracil. So these are pyrimidine bases. So we are taking that DNA which contains the purine bases. One is adenine, it has adenine, and the second one we are taking guanine, and that the third one it is cytosine, and the fourth one which has, that is thymine. It has thymine. So these four molecules is present in that DNA. What is that? Adenine, guanine, cytosine. These are common for both DNA and RNA. And here thymine is present in DNA. Instead of thymine, uracil is present in RNA. That is RNA. That is RNA which contains one. It is adenine, and it is guanine. We are taking guanine, and third one cytosine, and fourth one uracil. So these are the four nitrogenous bases present in that RNA. So RNA which contains adenine, guanine, cytosine and uracil, and DNA which contains adenine, guanine, cytosine and that thymine. And here adenine and guanine are purine bases, and pyrimidine bases cytosine is common in DNA and RNA, and another two, thymine is present in DNA and uracil is present in RNA. Now we may take that the structure of nitrogen bases. And nitrogen bases are two types. One is purine. We are take, it is a purine bases. And we are take the structure of purine. Now we are taking the purine structure. We are taking like this. That is here nitrogen. And we are take like this. And here N. And it is nitrogen base, that is a purine base, and we are taking here, there is the nitrogen, and here there is NH, and here the bond, and here the bond, and here the bond. Then it is a purine. We are taking that purine bases, and we are taking that the purine bases are adenine and that guanine. So here purine bases are, we are taking, one is adenine and another one guanine. So we are taking that the adenine, here N, and here we are taking like this, and here nitrogen and here nitrogen, then here NH, and here the bond, and here we are taking that the bond here, and in this adenine we are take adenine which contains NH2 groups, and we are taking that is guanine, we are take that is nitrogen, and here nitrogen, and it has NH, and here N, and we are taking here, and the bond here, we are take the bond here, and so here it has the CO group, double bond O, and here NH2 groups. So NH2 groups and CO groups are present like this. So here N, and here 1, 2, so here the bond, and 1, 2, so that here there is NH2 groups. So it is nothing but guanine. What is that? Guanine. So this structure of guanine, these two are made up of purine bases. Now we are taking that the pyrimidine bases. We are taking pyrimidine bases. Pyrimidine base. And we are taking the structure of pyrimidine only this hexagon. So we are taking that the pyrimidine is like this. That is nitrogen, and here that is nitrogen, and here the bond and this bond. And in this pyrimidine there are three types of bases. One is cytosine, and also that thiamine, and also that uracil. So here it has cytosine, we are taking like this, it has cytosine, and here nitrogen, and cytosine NH2 and CO group. So here that is NH2, and we are taking that here CO group. So there is a bond. So it is a cytosine. And the next one, it is thiamine and also that guanine. So we are taking that is a thymine structure. So we are take this thiamine, here the bond, and we are take double bond O, and here double bond O, 1, 2, 3, 4. So it is, and here it has a CH3 group. And here we are take NH, and here also here it has NH. So it is thiamine. What is that? Thiamine. And the next one, it is uracil. We are taking that the same structure like the thiamine. Here nitrogen, and here the bond, and here double bond O, and here double bond O, and here NH, and here 1, 2, 3, and that NH, 1, 2, 3, and it is NH. So it is balanced. So we are taking, it is uracil. So these are the structure of the purine and that pyrimidine bases. So we are taking here the purine bases are adenine and guanine, and pyrimidine bases are cytosine, thiamine, and uracil. And DNA which contains, we are taking DNA, and DNA which contains, what is that? Adenine and guanine, and also that cytosine, and also that thiamine. And we are taking that RNA and RNA which contains, that is adenine and guanine and cytosine, and instead of thiamine it has uracil. So these three are common in DNA and RNA. Now we are taking that is the pentose sugar. That is pentose sugar. And here pentose sugar in DNA it has 2-deoxyribose. D-ribose. So we are taking that DNA which contains 2'-deoxy-D-ribose. So this is the sugar which is present in that DNA. And if we are taking that RNA, it has only, that is D-ribose. So the D-ribose sugar which is present in that RNA. And in both cases, the D2-deoxy-D-ribose and also that D-ribose in RNA are in that beta Furanose structure. So both are in beta Furanose structure. So what is that beta Furanose? Beta Furanose is nothing but the 5-membered ring which contains oxygen. So it is a cyclic 5-membered ring. Cyclic 5-membered ring which contains oxygen. So this is the beta Furanose structure. So we are taking DNA which contains 2-deoxyribose. So we are taking 2-deoxyribose sugar. Now it has CH2OH and that here OH and here OH and that here there is a OH group like this. Now we are taking deoxyribose, that is DNA which contains deoxyribose. And deoxyribose, there is no oxygen, so oxygen atom is absent, then it is deoxyribose. And the structure is here 1' and 2' and 3' and 4' and that 5'. So these are the numbering. Now we are taking that is D-ribose. We are taking D-ribose which contains oxyribose. So here we are taking that here. Then we are taking CH2OH and here OH and here OH and here OH. Then it is D-ribose sugar. And what is the difference between deoxyribose and the ribose? And here there's 2' position, there is only hydrogen, and 2' position, there is a OH group. So here 1' and 2', 3', 4' and that 5'. So here it has OH group, then it is a D-ribose. And here only hydrogen, then it is deoxyribose. And here in a nucleotide, what is the linkages? So the phosphate sugar is linking with that 5' position of OH group or maybe the 3' position of OH group. So there is a phosphate linkage. So the phosphate linkage is occur in the pentose sugar at the 5' position or the 3' position. So we are taking that the DNA RNA which contains nitrogenous base and also that pentose sugar, and the third one it is a phosphate group. And phosphoric acid form a phosphodiester. So we are taking that it has the phosphodiester linkage. Phosphodiester linkage, and it has, we are taking, that is the phosphorus double bond O and O minus and O minus, and here it is O minus. We are taking, depending upon the phosphate group, depends on phosphate group, and the nucleotide which has classified into mononucleotide and dinucleotide and trinucleotide likewise. Now we may take that the nucleotide and nucleoside. So we are taking nucleoside and nucleotide. Nucleotide. So what is the difference between nucleoside and that nucleotide? And here, absence of phosphate group, then it is known as nucleoside. So the nucleoside which contains only, that is a pentose sugar and a nitrogen base. So we are taking the nitrogen base, and it has the pentose sugar, and without the phosphor linkage, then it is known as nucleoside. So it has a nucleoside. From that the nucleoside, we are getting the nucleotide when treating with the phosphoric acid. So we are taking that here when this nucleoside is made up of nitrogen base and that pentose sugar. And this nucleoside is reacted with the phosphoric acid, and we may get the phosphate linkage. Then the phosphate linkages is added in that nucleoside, then we are getting nucleotide. It has nucleotide. When the nucleotide on polymerization, we are getting polynucleotide, and is nothing but nucleic acid. So we are taking the nucleotide on polymerization. So it is on polymerization, then we are getting that it is polynucleotide. Then it is nothing but nucleic acid. Okay, so this is the difference between nucleoside and nucleotide. So nucleoside is made up of only nitrogen bases and sugar without a phosphate group. If phosphate group is added, then it is called as nucleotide. Then the nucleotide on polymerization, we are getting that polynucleotide, which is known as nucleic acids. We are take the pentose sugar here. This is the pentose sugar, and which contains here CH2 and that O. And phosphate group, it is the 5' position. Phosphate group is attached with that the 5' of pentose sugar, so that it is a sugar. Then here it has P double bond O and O minus and that O minus. And we are taking here there is a OH, and here the bond, and here there is a base, that is the nitrogen bases which is attached. And so it has the phosphate group and sugar and the bases, then these three are nucleotide. Now it is attached with one more, we are taking that here, we are taking this one. So another nucleotide. So we are taking the another nucleotide which has CH2 and O and phosphorus, that is phosphate group, O minus and that O minus. And here the bond, it may be OH, and it may be DNA or RNA, H or OH, and here there is a base. Here there is a base. And these two are attached. One nucleotide is attached with another nucleotide at that 5' position and maybe the 3' position. 5' position and maybe the 3' position. So 5' and 3' phosphate linkages which is occur. So here it is a 5' position, and here it is a 3' position. So it linkages these two positions. So here there is a linkages. Now we may take that. Here there is a oxygen, and which contains the base. It has a base, and here it has CH2 group and O and phosphate group, and here O minus and that O minus, and it has the 5' position. And another nucleotide may be linked at a 3' position, so that it has the 3' position. So it is linked with phosphorus. So O, and here phosphorus and double bond O and that O minus, and here O, and here it is CH2. Now we are taking like this. Then here there is a OH group and maybe H, and here there is a base. Now this is the linkage that has 3' and that 5' position. Double strand helical structure of DNA. And here in 1950s, Rosalind Franklin So Rosalind Franklin and another one scientist Wilkinson, there is Maurice Wilkins, used the X-ray diffraction to explain that the DNA structure. So they are used X-ray diffraction to explain the DNA structure. And after that, in 1953, Watson and Griggs given the three-dimensional model of DNA. So here in 1953, that is Watson and Griggs. Given the three-dimensional model. Of DNA. And here in the three-dimensional model of DNA, it consists of a strand, that is two helical chain is attached right-handed double helix. So the two helical chain is wound around the axis, around the same axis to give the right-handed double helix. So here these two helical chain, they are anti-parallel. So the two anti-parallel chains is wound around the same axis, and that will be produces the right-handed double helix. And here taking the hydrophilic group. So hydrophilic group, one is, that is the sugar, which contains OH group, and the second one, it is the phosphate, which is a phosphate. So it has O minus group. So the hydrophilic groups are present in outside of the helix. It is present in outside of the helix, and it is surrounded by water. So we are taking the three components, that is sugar and phosphate and nitrogenous base. And this sugar and phosphate are hydrophilic group, which is easily attached with that water molecule, so that they are present outside of the helix. Now we are taking that the nitrogen base is present in that inside of the helix, which is stacked. So nitrogen base is stacked inside of the helix. So here we are taking that nitrogen bases are present in that inside of the base, inside of the helix, and on the outside there is a sugar and that phosphate. And here we are taking that the nitrogen bases which contains the cyclic ring. And these cyclic ring are hydrophobic character. So the nitrogen bases are hydrophobic. That is hydrophobic. So hydrophobic nitrogen base is present in the perpendicular direction. So it is present in perpendicular direction. Perpendicular direction. So it will be present in a perpendicular direction so that it will be reduces the repulsion between that the phosphate and that nitrogenous base. And it will be produces the major and the minor group of that duplex. So it will be produces the major and minor group. Major and that minor group of duplex or helix. We may take that the structure of double helix structure of DNA, and this is the double helix structure. Double helix structure, and we are taking that this turn. Each turn which contains 10.5 bases. So each turn we are take 10.5 nitrogen base, and it has the length 36 angstrom. So here there is a length 36 angstrom which contains, and we are taking the length of this nucleotide. So the nitrogen base nucleotide, it has only, so we are taking that the base as the length only, it is 3.4 angstrom. So it is the length between these two helix, nitrogen base. Now we may take that here the outside. Here there is a outside of the helix. Outside of the helix which contains sugar and that phosphate. Sugar and that phosphate, they are surrounded by water. They are surrounded by water, and it has a hydrophilic character. It is a hydrophilic character. Now, inside there is a nitrogen base. So, it is nitrogen base. So, the nitrogen bases are stacked inside. You know that the nitrogen bases. Now, we may take the complementary pattern. We are taking the complementary pattern. So, what do you mean by complementary pattern? Here, adenine is always bonded with that thymine. Adenine is always bonded with the thymine by that hydrogen bond. We are taking that hydrogen bond, and here there are two hydrogen bonds present between adenine and thymine. So here A is always jointed with that T by two hydrogen bonds. Now it is a complementary pairing, and we are taking another one pairing, that is guanine and cytosine. So we are taking, it is the first one. Now the second one, it is guanine. Guanine is always paired with that is a cytosine. So it is a cytosine, and by making, that is a 3 hydrogen bond. 3 hydrogen bond. So here it is a complementary pair, and by making that hydrogen bond, if the complementary pair is different, and which will destabilizes the helical structure. So helical structure is made up of the forces. One is a hydrogen bond, and that is a stacking base pair interaction. So we are taking the stability of helical structure. Stability of helical structure. So the stability of helical structure is made up of hydrogen bond of complementary pair. Hydrogen bond of complementary pair. That is AT and GC, and another one that is stacking interaction. So we are taking that is nitrogen base stacking interaction. So stacking base pair interaction. Stacking base pair interaction. And this stacking base pair interaction is non-specific. It is a non-specific, and it will be take the major role of the stability of a double helix. So it is the structure of DNA. Now we are taking the types of RNA. There are three types of RNA. One is rRNA. rRNA is nothing but ribosomal RNA. Then the second one, that is tRNA, that is transfer RNA. And the third one, we are taking mRNA, that is messenger RNA. Messenger RNA. Now we may take this function of these three types of RNA. The first type, that is ribosomal RNA. Ribosomal RNA. And the ribosomal RNA present in that cytoplasm and that chromosome. So it is present in cytoplasm. It is present in cytoplasm and chromosome. And that it is a chromosome. And which contains 60% of rRNA and also that 40% that is protein. So it has 60% of RNA and 40% of proteins. 40% of proteins. Now it will be, so the synthesis of nucleic acid is done on this site. So it will be produces the sites synthesis of nucleic acids. Synthesis of nucleic acid. So it will be produces the sites on synthesis of proteins or nucleic acids. Then the second one we are taking that is a transfer RNA. Transfer RNA. And this transfer RNA has lower molecular weight. This is lower molecular weight. And a single strain which contains 74 to 93 nucleotides. So we are taking a single chain which contains 73 to 94 nucleotides. And here this transfer RNA carries amino acids to that protein synthesis sites. So it carries amino acid to protein synthesis sites. Protein synthesis sites. So it will be carries the amino acids so that it will be taken as a transfer RNA. Now we are taking that the third one, that is a mRNA. And if you are taking that the mRNA, and mRNA is a single strand. So it has a single strand. And there are two processes, and there is a transcription and a translation that will be done on that mRNA. Now we may take that this mRNA is synthesized from that DNA. So mRNA is synthesized from DNA. Then it is known as transcription. So the process which is known as, it is a transcription. So transcription is nothing but mRNA is produces from the DNA. And the second process we are taking that here the mRNA carries the genetic information to, that is from that DNA to, that is nucleic acid. Now we are taking here that is mRNA, it carries genetic information from DNA to chromosomes. So we take that is chromosomes. Then the process which is known as that is translation. So transcription and translation that will be done on this mRNA. So in this session we are taking that the structures of DNA and the structure of purine bases and pentose sugar and also the phosphate, and also we are taking the types of RNA. Thank you.